So for this particular study on digoxin, we did two sets of analyses. We did first what we call baseline analysis or prevalence analysis. So these were patients already taking digoxin when they entered the trial. The second analysis is what we call incidence analysis. So in other words, these were patients that were not using digoxin at the beginning of the trial, and they started digoxin during the course of the trial. So in our first analysis, the baseline, we did not show an association between digoxin use and mortality. However, when we look at the levels of digoxin in the blood, the concentration of digoxin in the blood, we found a gradient of risk according to the levels of digoxin. In other words, for patients who had less than 0.9 nanograms per ml of digoxin concentration, there was no increased risk of death. For those patients where the concentration was between 0.9 and 1.2, there was a 16% increase of death, however not significant. But for those patients who were above 1.2 nanograms per ml, we found a 56% increase in the risk of death, and this was highly statistically significant. So again, we did not find an overall association in the baseline analysis between digoxin and death, but that's because most patients were below 0.9 nanograms per ml in terms of digoxin concentration. When the concentration was actually above 1.2, there was an important increased risk. And then when we look at the digoxin concentration as a continuous variable as opposed to different cutoffs, we found a 19% increase in the risk of death for each 0.5 nanograms per ml increase in serum digoxin concentration. So a really linear and direct association. The higher is the concentration, the higher is the risk of death. For the incidence analysis, then when we look at the patients who are not using digoxin and started using, then we found a 78% risk of all-cause death in patients starting digoxin, and this was highly statistically significant. Importantly, we look at patients with and without heart failure, and we saw a similar pattern, that starting digoxin was associated with higher risk in both patients with heart failure and without heart failure. The risk was even higher in patients without heart failure, with a twofold increased risk. And finally, we look at sudden death, because the most common cause of death of these patients was sudden death. And when you look at sudden death in patients starting digoxin, there was a fourfold increase in the risk of sudden death compared to patients who never started digoxin. Interestingly, most of these sudden deaths occurred in the first 6 months of the initiation of digoxin therapy.